All right, so today we're going to learn Chapter 5, Energy and Life. So we're going to talk a little bit about tonicity to start off. Then we're going to talk about what energy is. We're going to look at the structure of ATP. And we're going to talk about activation energy, enzymes, things that affect enzyme action, and then lactase, which is just a type of enzyme. All right, so for cells to survive, they must be able to regulate water movement across their membranes, whether it's in or out of the cell. When we talk about tonicity, we're actually describing the solution that the cells are in. So in our case, it would be, our blood, if we're talking about our blood cells. So our plasma, all of the non-cellular components of our blood would have some kind of tonicity. So when we're talking about the tonicity of any kind of solution, we can ascribe different names, 3 different types of names to it. So we can have hypotonic solutions, we can have isotonic solutions, and we can have hypertonic solutions. So hypo, to break down hypo, it actually just means below or lower than. So if your solution is hypotonic, that would mean that the solute concentration, I forgot to mention. So your tonicity refers to the solute concentration, so the amount of stuff that's dissolved in the solution. So if we're just dealing with water, for example, your solute could be things like sodium or chloride or sugars, just stuff that's dissolved in the water. And so if you have a hypotonic solution relative to a cell, that means that the solution you're talking about has less solute in it than the cell does. Now, if we have an isotonic solution, that means that the solute concentration is equal within the solution to within the cell. And now if we're talking about a hypertonic solution, you have more solute, a higher solute concentration outside of the cell than you do within the cell. So here's a solution here. We'll say that this teal area is the solution, and then the green dots are solute. And so you actually have a higher density of solute outside of the cell than inside of the cell. So we would label this hypertonic. All right, so we have like a quick little quiz based off of what we know about how water moves in and out of cells, or how water moves through semipermeable membranes. We should be able to infer what happens if your cells are in these various types of solutions. So if you place a cell in a hypotonic solution, the solute concentration is lower outside of the cell. So what do you think is going to happen to the cell itself? So we know that water has a tendency to move to where there's more solute, and so you're going to have water on the outside of the cell with less stuff in it, with less solute in it, and it's going to want to move into the cell where there's more solute because it's a hypotonic solution. So there's more solute in the cell than there is outside, and so what happens is the water will travel through your cell's semipermeable membrane and cause it to swell up. Your cell will actually get larger. Now, if you swell up enough, you might undergo a process called osmotic lysis. So lysis means to cut. If we're talking about red blood cells, we call this hemolysis or hemolysis. Now, if you've got an isotonic solution, that means you've got the same amount of solute outside of the cell as you do inside of the cell. What you're going to experience, what your cell's going to experience, is a net zero gain of water, or a net zero movement of water. So you're going to have an equal amount of water moving into the cell as you are going to have water moving out of the cell. And so you're just going to break even. Cell volume will not change, water is at a dynamic equilibrium. Now if you have a higher solute concentration outside of the cell, so if the solution is hypertonic, what's going to happen is all of the water within your cell is going to want to move out to dilute the solute on the outside of the cell. And so as a result, your cell will shrink, your cell will lose water and shrink. That's a process that we would call crenation. Okay, so we were talking about our red blood cells. Hypotonic, you get the hemolysis. Isotonic, nothing happens. You get the shriveled cell if it's in a hypertonic solution. However, when we're talking about other types of eukaryotic cells with cell walls, such as plant cells, they have on the outside of their cell membranes cell walls. What these cell walls do is they actually allow for more rigid structure, and they actually protect the cell from bursting, for example, if it is experiencing or in the midst of a hypotonic solution. So if you put a plant cell in a hypotonic solution, your cell wall, or the plant cell wall, will actually prevent it from bursting. And so what happens is it swells and it just becomes turgid. So this is kind of like your crispy iceberg lettuce. Now, if you have an isotonic solution, we consider the cell to be flaccid. There's no net exchange, or there is an exchange, but there's no net gain or loss of water. And so this is kind of, I guess, a less crispy iceberg lettuce. Now, if you have the plant in a hypertonic solution, so let's say you just put it in a bunch of salt water, what happens is all of that water, all that fluid from within your cell is going to leave to dilute that solute. And as a result, your plant cell will actually shrivel. However, the cell wall maintains its structure. It maintains its structure in a relative sense, where it doesn't actually follow suit with the plasma membrane. So what you have here is a retraction of the plasma membrane, and this is something that could easily be seen under the microscope, and I think embedded in this PowerPoint there's actually a video showing that your plasma membrane will retract away from the cell wall, however the cell wall will stay in place. And so this would be an example of, like your wilted iceberg lettuce. I think this might be, oh no, this is just showing that. So normally in lab, what we'll do is we'll look at Melodia, which are just a type of plant in water. And these little green guys here, those are actually chloroplasts moving around. Now, if you were to put this slide, for example, or if you were to take some salt water, some high concentration salt water, and drip it onto this slide, for example, what you would notice is that the membrane would actually scrunch in towards the center of the cell, and then these little brick-shaped cell walls would remain in place. And I think this video actually shows it. Okay, so here's the summary. A lower solute concentration outside of the cell. So if you're in a hypotonic solution, meaning you have lower solute concentration outside of the cell, that means that the cell will burst or swell or grow, get larger. Isotonic means that the solute concentration is the same on the inside as the outside of the cell will remain the same size. In a hypertonic solution, which means you have a higher concentration of solute outside of the cell, then the cell will shrink or shrivel. Okay, so which figure depicts an animal cell placed in a solution hypotonic to the cell? So hypotonic meaning you have a lower solute concentration outside of the cell, meaning that the water is going to want to move into the cell. And it's also an animal cell, which means that this cell does not have a cell wall, meaning it might burst. So A would be the correct answer. Water is moving into the cell, and the cell is bursting. This would be an example of an isotonic solution. This would be an example of a hypertonic solution. And this would be an example of a plant cell in a hypotonic solution, because it only shows water going in. All right, so let's talk about energy now. So energy, in short, is just the capacity to perform work or move something against some force. So we've got some energy types. We have potential energy. So that's something that could be stored in chemical bonds. And when it's used, it can be turned into kinetic energy, or it could be converted into another type of potential energy. And then you have kinetic energy, which is just energy in motion. So this ball on top of the hill has a lot of potential energy. You move it down the hill, and what you're doing is you're converting this potential energy into the energy of motion, into kinetic energy. Now, this ball is a great example when we're talking about, like the macro picture, but when we're talking about the micro picture, we actually store chemical energy within bonds. So for example, your fat molecules or your sugar molecules, those bonds, those carbon to carbon bonds, store a lot of potential energy. And so here's a pretty good depiction. So as this coaster moves, we'll start from the beginning here. So it moves up, and it has more potential, and then it's converted into kinetic, and then it's potential, kinetic, and then, so you can see the correlation. All right. So here's kind of a comparison between, two different things that could actually convert chemical energy into kinetic energy, or just another type of energy. So if we take our car, for example, we use gasoline, which in most cases have a lot of carbon. So these little black things here are carbon. So carbon-carbon bonds. In those bonds, you store a lot of energy. We combust it, and as a result, we combine it with oxygen. It combusts, and we yield water and CO2. And what we're doing is we're converting this potential energy in this gasoline to the kinetic energy of movement. And that's what we're doing for, when we're driving our cars. Well, the same thing applies to food. So this looks like probably a triglyceride. We have these carbon bonds. We break that down within our cell through a process called cellular respiration. And what we do is we actually convert that not only into kinetic energy, but into ATP. It's just another potential energy. So we're converting this potential energy into another potential energy, and we use that potential energy to actually make the cell do stuff. In the process, and this applies to both of these examples, in the process we lose some of that energy, that potential energy, to an unusable heat. So your car doesn't really do much with the heat. Our cells don't really do much with the heat. But in the end, we're still getting CO2, water, and then in this case, instead of converting it into kinetic energy, we're just converting it into another type of potential energy called ATP. So let's talk about the laws of thermodynamics. The first law is that energy cannot be created or destroyed. It can only change form or move transferred from one object to another. So I have a typo there, sorry. So energy can't be created or destroyed. You can only change its form. And so, in this case, you have potential to kinetic. It's converted from potential energy to kinetic energy. You can convert from potential energy to potential energy, just a different formulation of potential energy, a different type of potential energy. The second law is as energy is converted from one form to another, some of it is converted into a form that is unusable. So heat is usually that unusable form. So poor efficiency, unable to do work. We don't really use heat to do work for us. So an analogy that I thought of here is, let's say, we're in India and we have a bunch of one type of currency. So we have a lot of US dollars, and we need to get rupees so that we can actually use, a currency while we're in India. In order to convert our US dollar into the rupee, what we do is we have these things called exchange fees. Usually the person converting that money for you will actually take a small fee, and we just kind of like lose it to the abyss. Same thing kind of goes so this is the analogy for this. So we eat a bunch of food. We can't directly use all of the carbs, the sugars, or the sugars, the proteins, the fats, in this tikka masala. What has to happen is we need to convert this potential energy into the ATP. a usable form of energy, potential energy. And in the process, we lose some to heat, an unusable form. Okay, so let's talk a little bit about entropy. So entropy is just, it translates roughly into disorder. And what I'm saying here is that life will require a constant input of energy to maintain order. So in order to move from this space, this room here, to this room, to maintain this order, this takes a ton of energy to do. However, to move from this type of room to this type of room, usually it's going to happen spontaneously. Not very much energy is needed to make this happen. This is something that just happens naturally. So all cells need ATP to capture, transfer, and store energy. In order for our cells to maintain order within themselves, they need ATP, which means they need to harvest whatever kinds of things we eat for that ATP. Okay, so ATP, adenosine triphosphate is what the acronym stands for. Adenosine tri, so 3, phosphate, 3 phosphates. Okay, and so this is just going to be the energy currency of all of our cells. You got your adenine, you've got your 5-carbon sugar, and then you have 1 phosphate, 2 phosphate, 3 phosphates. Now you have very high energy bonds in your between your 3rd and 2nd phosphate, and between your 2nd and 1st phosphate. So these are very high energy, and by what I mean by high energy is that when these bonds are cut, for example, a lot of energy is released. Also, these bonds here are very unstable. So they really want to, let go of this phosphate. And so they're going to let go of this phosphate very, very easily, pretty much spontaneously. However, between this phosphate and your sugar, not too high energy. So you have highest energy here, your highest energy bond is here, and your second highest energy bond is here. And so let's say we lose a phosphate, so we're adenosine triphosphate, we lose a phosphate very easily. We are now called adenosine diphosphate, di for 2. If we lose this second phosphate, now we're adenosine monophosphate. So pretty simple. Okay, so ATP, what it does is it releases energy when it transfers a phosphate group to another molecule. So you go from ATP to adenosine diphosphate, plus that phosphate, wherever it is, plus energy. So you release some energy in the process. Okay, so which of the following statements about ATP is true? Let's start from the bottom. It releases energy when 1 phosphate group leaves ATP. Oh, well, that's the correct one. It contains a sugar group glucose. That's false. It contains ribose. Extremely stable bonds link the 2nd and 3rd phosphate groups. Well, we just talked about this. These are actually extremely unstable bonds that link your 2nd and 3rd phosphate groups. And it contains 5 phosphate groups. It actually contains only 3 phosphate groups. It contains 3 phosphate groups, adenosine triphosphate. Okay, so let's talk about activation energy. Or let's just talk about energy needed to start a reaction. So the energy needed to start a reaction sometimes, or the energy needed to start a reaction is the activation energy, and sometimes the energy or the activation energy for reactions may be different from others. So to move from your reactants to your products, you must overcome this hill. So this is an analogy for activation energy. So in order for these reactants to move from their original state to where our cell wants them, some energy needs to be pushed into them to move them over this hill. Once they get over this hill, very, very, very easy to just drop on down, and it's almost spontaneous from that point forward. So what enzymes do is they catalyze or lower the activation energy. So your original hump here, or your original hump was this blue line here. So this was the original activation energy for the reaction. And that's if you're trying to just move from, like one molecule type to the other with no helpers. Now, if you introduce an enzyme, what it does is it lowers the energy needed in order for you to get from, one molecule type to its product type. So all enzymes are generally protein catalysts. Generally, they're composed of proteins. And they're just catalysts that get reactions started quickly. The way they do that is they lower the activation energy. So here's your enzyme. Your substrate is the stuff that is going to be modified by your enzyme. And your enzyme is going to bind to the active site of the enzyme. Okay, so enzymes are mostly proteins. And so this looks like an enzyme that is, what it's doing is it's combining two different things to make one bigger thing. Your active site is where the substrate will bind. So your substrate, let's just say, what are two things that we would want to combine? Let's say glucose and fructose to make sucrose. So we want to make sucrose. So you've got glucose here, which is going to bind to an active site, its active site. And then you're going to have fructose here, which is going to bind to this other part of the active site. Now, once they bind, what you get is a enzyme substrate complex. So it's just the enzyme complexed with its substrates. And then the enzyme does its thing. So it lowers the activation energy for the reaction. It helps conduct the reaction. And you combine both your glucose and your fructose to make sucrose. So here's your larger product. So what happens here is, well, your enzyme, its three-dimensional shape is what actually dictates what kind of reaction it's going to do. So this enzyme can only combine glucose and fructose to make sucrose. It can't do any other type of reaction. You can't combine any other things. It's not going to break down anything. It's just going to stay here and not stay here, but it's going to only conduct one type of reaction. Now, after that enzyme is completed with its job, what it's going to do is it can be reused. So it's not going to be depleted after the reaction. So there are two major factors that will affect your enzymatic action. That's going to be temperature and pH. So each individual enzyme will have a sweet spot for what temperature it prefers and what pH it prefers, what pH it will work optimally in, and what temperature it will work optimally in. So optimal temperature and pH will vary depending on what type of enzyme you're talking about. So let's say, we've got this chart. The x-axis is your temperature in Celsius or centigrade, and then your y-axis is the rate of reaction. So let's say this blue enzyme here, it functions best at 4 degrees Celsius. So this would probably be an enzyme that we would find in a colder climate. So let's just say an enzyme that lives in a bacteria, or an enzyme found in a bacteria that lives in the North Pole. Now here we got 37 degrees Celsius. So this could be like an enzyme that you'd find in your own body, or an enzyme that you'd find in a bacteria in your gut, for example. And then here you have, your green enzyme, its sweet spot's pretty high in temperature, almost boiling. And so you'd probably find, this enzyme living within a bacteria living in a much hotter climate. So despite all this, extreme temperature and pH what it'll do is it'll denature an enzyme. So to describe what denaturation is, denaturing something is to kind of break it down, to make it a gooey, sloppy protein. So if we're denaturing a protein, what we're doing is we're breaking apart its hydrogen bonds and even some of its covalent bonds that actually hold it together. So what it's going to do is it's going to change its shape. If we're breaking those hydrogen bonds, we're changing its 3D shape. Remember, its 3D shape, an enzyme's 3D shape is what determines its function. And so if we change its shape, now it can no longer function. And so let's say this enzyme that we find in a bacteria living in the North Pole, we bring it to a more temperate climate. is actually too hot for that enzyme, so it becomes denatured. So the same thing would actually apply to pH. We have a sweet spot, all of our enzymes in our body have a sweet spot for pH. So, for example, the enzymes that hang out in our stomach to break apart, to break down and digest food, probably work optimally in an acidic environment, whereas maybe the enzymes that we secrete, onto the surface of our skin or that we'd find in our digestive tract might actually be optimally, they might perform optimally at a slightly higher pH. Okay, so some things that can interfere with enzymes are inhibitors. So we've got two different types of inhibitors that we'll talk about in this class. We have competitive inhibitors. What these do is they bind to the enzyme's active site to block substrate from binding. So you have substrate, which typically binds to the active site. And what happens here is if you have a competitive inhibitor, what it's going to do is it simply binds there, blocking the substrate that would normally bind there from binding. So you can't get a reaction at this point. Now we also have something called non-competitive inhibitors. So what they're going to do is they're not going to compete with the substrate for binding to the active site. What they're going to do is they're going to bind to somewhere within the enzyme, usually called an allosteric site. So it's going to bind to an allosteric site on the enzyme, and what that does is it changes the conformation of the active site so that the substrate doesn't even want to bind. It can't fit. So those are non-competitive inhibitors. Competitive inhibitors compete with your substrate. Non-competitive inhibitors will bind to another site on the enzyme to change the conformation of the active site to prevent the substrate from binding to it. So when an enzyme catalyzes a reaction, what's going to happen here? Let's start from the bottom again. It is used once and discarded. False. So enzymes can be reused over and over and over again. When an enzyme catalyzes a reaction, the enzyme acts as a reactant. So a reactant is something that is going to be converted into something else. If we're talking about the case of the example that we used, where we had the glucose and the fructose combined to make sucrose, those were the reactants. The enzyme itself is not a reactant. When an enzyme catalyzes a reaction, it raises the activation energy of the reaction. It's actually the inverse of this. It lowers the activation energy of a reaction. So, must be A, it lowers the activation energy of a reaction. So let's talk a little bit about lactose. Lactose is a sugar because it ends in ose. Your sugars will end in ose. Glucose plus galactose will make lactose. So that would make this a disaccharide. So lactose is just a sugar that makes up about 2 to 8% of milk. And what it does is it enhances your absorption of calcium, magnesium, and zinc. Lactase, on the other hand, it ends in ase. So this means that it's an enzyme. Typically, your enzymes are going to end in ase. If you ever see something ending in ase, typically means that it's going to be an enzyme. You're talking about an enzyme. And you can kind of infer what it does based off of its name, So lactase, likely this is going to break down lactose. So lactase is a mammalian enzyme that we usually find in the villi of our small intestine. So this is something that's going to break down lactose. We find this on chromosome number 2, and it's going to work best at pH 6, so slightly acidic. So lactose intolerance, when you have lactose intolerance, that means that you've stopped producing this enzyme. So the lactase enzyme sadly begins to decrease in production and expression around the age of 2. So this is something that's actually been genetically programmed. It's a genetically programmed loss of function. So typically, around the age of 2, little humans begin to become weaned off of their mom's milk. And so what's the purpose of spending energy to express the gene associated with lactase if we're actually no longer, drinking our mother's milk after the age of 2. So that's kind of why it's genetically programmed loss of function. And so as a result, we become lactose intolerant. Many of us do become lactose intolerant. And all that is the inability for us to break down lactose into glucose and galactose. So lactose intolerance before age 6 is usually rare. So we begin decreasing at age 2, but it's not, it's not like a one and done. It's not like we get a sudden dip. It's just a gradual reduction. And so here is a map showing the incidence rate of lactose intolerance around the globe. And so if you notice, the darker it is, the more incidences of lactose intolerance you'll find. So as you go south of the equator, you actually have a high incidence rate of lactose intolerance. And as you go north, with this is an exception, but if you go north of the equator, what you'll notice is this northern European area actually has the lowest incidence of lactose intolerance. And what we call this is lactose tolerance. And so this is a question for you to think about till you watch the next video. So why do you think lactose tolerance mutation could have spread through the population of Northern Europe?